Okay, so chapter 9, the endocrine system. Kind of a small chapter, but big ideas. This is how we make proteins. And this relates back to all the rest of the genetics we've talked about before. Remember the whole Punnett square and whatnot? So you got these genes that you inherited. What do you do with them? Those are the instructions for making proteins. So we're going to talk about how to do this. And there's this idea in here of the central dogma. It sounds very self-important, but here it is. So you got your DNA, that's the genes. Um, that's used to make RNA. Doesn't turn into it, it just is used uh, as a code to make more code, both in the nucleic acid language. And then at a ribosome, we switch to the protein language and actually make something that will do something in our body. That is gene expression in a nutshell. So this protein, that can be a hormone. It could be the receptor for the hormone. It can be structural proteins, like the hair on your head, some of the keratin in your skin. It could be muscle protein. It could be collagen in your skin, in your bones. There's thousands of types of proteins that we make, and each is coded for by a unique gene. Enzymes as well. So any protein we make, we need a DNA code, and here's how it's expressed. So let's jump into that. All right, so we're going to spend quite a bit of time on the gene expression page. This is what it's all about. First, in terms of context, this is stuff down on the nanometer scale again. This is molecular genetics. Let me actually write that. Looking at the actual molecules of genetics, the DNA, the RNA, the proteins. We've actually done some molecular genetics before when we did DNA replication. So this has some similarities, but then goes well beyond it as well. Let's first put this in the context of inheritance. So sperm fertilizes an egg. They each carry alleles. So let's say it carries a recessive and a dominant allele. Those comes together in fertilization. And so we have a heterozygote. Well, let's switch codes a little bit. These were the sort of codes we used when we filled out Punnett squares. But what is that? Obviously, there's no capital H inside your cells. And there are not a bunch of A's and T's and G's and C's either, but there are a bunch of nucleic acids, the DNA, right? Each gene is made of DNA. And that DNA is made of nucleotides. So the sequence is really important. So there we go. The gene and the allele is a sequence of nucleotides. That's the instructions. So if you compare these two, we've got dominant, recessive. How does this one stack up against the other? You can see they're pretty similar. It's this part right here, that's the difference. So even just one nucleotide change can change the outcome. What do I mean by the outcome? Well, let's take a look at that. All right, well, let's start with transcription. So some of the more detailed view on the right, looking at it in the cell context in the left. This happens in the nucleus. Of course, this is because our DNA, in this case allele capital H, the dominant allele, is there. So that's where we have to do transcription. In the end, what we want is messenger RNA. So it's another nucleic acid made of a bunch of A's and T's and G's and C's, this thing over here. To get there, we need to use an enzyme, and that enzyme is RNA polymerase. Should sound familiar, just like DNA polymerase, and it does the same thing, except instead of writing DNA, it writes RNA. And the RNA code is pretty similar to DNA. Remember, in DNA we had the nucleotides A, T, G, and C. For the RNA, we also have A, but instead of T, we have U for uracil, and then also guanine and cytosine. So same thing, we just need to make a substitution. If we would have put a T, then we put a U. There we go. So this process of transcription, first, in initiation, the RNA polymerase lands at this promoter, and it starts going from the 3' end to the 5' end on the coding strand of DNA, laying down complementary bases. That should sound pretty familiar. So it's taking all these bases here, these free RNA nucleotides in the nucleus, and laying them down one at a time, one after the other, matching up the complementary bases to the gene, the DNA. Once we're done with that, DNA is in two parts, right? So here's one strand, here's the other strand. That's going to stitch back together and stay in the nucleus. That's the whole point of this. We don't want to clip a gene out and take it out of the nucleus. We just want a copy of that so we can use it. All right, easy enough. That's transcription, complementary base pairing. Each cell is going to make lots and lots of different proteins using lots of genes. Now in eukaryotes, but not in prokaryotes like bacteria, we also have to do this process where the RNA matures. So RNA maturation, this is kind of like a graduation for it. We do a couple of things. First, it clips out these introns, bits of DNA that don't actually code for anything. Actually, in this case, I guess it's RNA by now. So we cut out the introns, they stay in the nucleus. The rest of these guys down here we could call exons. So that's the good stuff, that's the code, and it's going to exit the nucleus here in just a minute. We also add a guanine with several phosphates, that's known as the 5' cap. And this end has a whole bunch of adenines. This is the poly A tail. I don't know, weird names in all this molecular genetics business. So that's the poly A tail. Both of those keep it stable. Otherwise it would get broken down right away. So there, at least in a nutshell, is transcription. So moving on now to translation. This is once that mature RNA leaves the nucleus through one of those little nuclear pores and finds a ribosome. So this right here in the background is a ribosome, and so is this. So we're going to assume we just have a single ribosome. There are free ribosomes, and there are some on the endoplasmic reticulum, but let's just keep that out of it for now. So what do we need? We need the messenger RNA to land on a ribosome. We need the two halves of the ribosome, the larger half and the smaller half, these are sometimes called subunits, to clamp onto the messenger RNA. And then we need to start by putting a tRNA, a very specific one, in place, and that starts the process. So there's initiation. Once all that's together, then the elongation phase happens where we just keep bringing in tRNA after tRNA into the ribosome. And they drop off their amino acids. So each of them carries this amino acid. We can see it over here. Same circles, but here I've drawn in the names, or at least the abbreviations: methionine, proline, tryptophan. They go into the ribosome. Then you have kind of two parking spots. And so only two tRNAs can fit in there at a time. How do we know which one is the correct one? Same rule as usual, complementary base pairing. So each of these is complementary to the messenger RNA. If that happens, then the ribosome confirms this is correct, this is a match. And then we need to add some energy from GTP, and a bond is made. So that bond links the amino acids. This other one gets broken, and so that tRNA amino acid bond gets broken and it's released. This tRNA can then float off and go find another amino acid, but these are now linked in a chain. And then this one shifts over and the next one comes in, and that process repeats. And that keeps going until it reaches a codon somewhere down here at the end, usually after several hundred amino acids, that basically tells it to stop. And then it's done. Everything falls apart and the protein is now made. So we got a whole chain of amino acids, and we need to fold that into the right configuration, because remember, for proteins, shape determines its function. And once it's folded, it can go do its job. One side note, we have this thing called a codon chart that we have used to make proteins. That basically does the job of a ribosome. So the cell does it with the ribosome. We can do it with the codon chart because we can read. Cells can't read, we can. So I guess we got one up on our cells. They just do it a lot faster than we do. So let's say we have a protein that we have made, but we want to get it out of the cell. So that requires a little organization. This does not happen in prokaryotes like bacteria, but in eukaryotes like us, plants, fungi, then we have this endomembrane system to help us organize all of the proteins that we make and get them out of the cell through exocytosis. So we can see this endomembrane system in two places in the concept map. I'll start here. So we talked about translation happening at a ribosome, and a free ribosome usually makes proteins that are released right into the cell to be used inside the cell, or I should say the cytoplasm. But if we wanted to export that protein, so here are these proteins, the little W shapes, and I'm exporting that out of the cell by a process known as exocytosis, then I need to put it in a vesicle. So that's this right here. All right, so how does that happen? I want to package a bunch of them at once so I can release them. Let's say it's insulin, and I want to release one insulin molecule. I want to release several. So we're going to start up here in the rough endoplasmic reticulum, this series of folded, twisted membranes, all in squiggles right by the nucleus, and they're studded with ribosomes. That's what all these little dots are about. And so that's where the translation happens. Since it's associated with membranes, the proteins don't drift off, they stay there and they get packaged in this little vesicle. That then goes to the Golgi apparatus. This guy right here looks like this little stack of folded pancakes, a series of membranes. There the proteins get modified, chemically tagged, and said, kind of like a post office, you're going to leave the cell. And so now they get put in another vesicle which merges with the membrane and goes out by exocytosis. So there it is there. Let's look at it in the context, just briefly, of the entire cell. So here's the entire cell. We made our RNA in here. It leaves into the endomembrane system, so it has lots of dots in this area. So that is the rough endoplasmic reticulum, which is different than the smooth endoplasmic reticulum, which does not have ribosomes. We make that, we put it in a vesicle, and that vesicle has to move. It doesn't just float off by itself, it has to actually be powered to move, and that's the job of the cytoskeleton, these little fibers right here. So it actually hitches a ride, goes down south in the cell. So here we are, it takes a ride down to the Golgi apparatus, gets processed, spit out the other side as a whole bunch of chemically tagged proteins that are destined to go to the edge of the cell. And so the process of exocytosis, where we get all those out, first that vesicle is going to move really close to the cell membrane, but the cell membrane is still intact. These are big proteins, so we can't just open up a big hole or a channel to get it out. So what we're going to do instead is that vesicle is going to merge with the membrane. So think of this kind of like step 2 in a little comic strip. So that's still filled with lots of proteins. So that is merged with the membrane. And then the membrane can sort of flatten out. Oops. And those proteins can exit the cell. And through steps 1, 2, and 3, think of that as a time series, we never broke the membrane. So we didn't ruin the integrity of the cell. So that's how exocytosis works. And this works for all kinds of proteins, whether it's a hormone, a digestive enzyme, or something else. That's how we get proteins out of the cell. The opposite of that, by the way, is endocytosis, which things like white blood cells use to get big particles into the cell. And if you just played exocytosis in reverse, then you'd essentially have endocytosis. So there's a preview of something else. In the context of the endocrine system, then we send out hormones to help regulate the body. But those hormones go all throughout the body, right? Because they're going through our circulatory system. So how does each organ know, am I supposed to respond? Answer, it has a receptor. Once that hormone hits the receptor, then there's a whole chain reaction of events that happen inside of the cell in order to get that cell to respond in a very specific way to fix a problem or regulate the body. Here's how that works. When we talk about cell signaling, I mean some other cell, whether that's a nerve cell or an endocrine gland, has released a chemical, and that chemical is meant to signal this cell, hey, I want you to do something. And often that will be activating an enzyme to make some other sort of chemical. The key thing is we need to have a receptor. So they don't really look like these radio antennas or satellite dishes. They're proteins embedded in the membrane. So they permanently sit outside on the membrane, and they are very specific. So insulin has its own receptor, glucagon has its own receptor, epinephrine, norepinephrine, you name the signal molecule, there is a specific receptor. So only if it has that receptor will it do something. One model for this is the G protein coupled receptor, where this receptor protein is modeled to, or is coupled to this G protein. That activates another enzyme, which then creates lots of other molecules that get released into the cell. Often those activate yet another enzyme, which then amplify the signal even more, because enzymes just keep doing chemical reactions until you shut them off. And eventually the enzyme we want, this target enzyme, gets activated through that process. So you may recognize this moving from one molecule to another one, to another one, to another one, as a signal cascade or even an enzyme cascade. We've talked about enzyme cascades before, this idea that they work in teams to get a more complex job done. And so this is another example of that, just in a different context. And so that's how cell signaling gets done at a basic level.